3, 4.
This one is a developing story as well. April 16th, 2024. It's not really anything to be like worried about right now. It's spawning a lot of interesting articles. It spawned the name Astrobiodefense and is a completely real, not hypothetical or fictional field of study. And this is from yesterday on space.com in response to this April 16th discovery that I'm about to read. But multi-drug resistance bacteria found on International Space Station mutating to become functionally distinct. In a new scientific paper funded by a Maze Space Biology grant, principal investigator Dr. Kasturi Venkataswaran of NASA's JPL Jet Propulsion Laboratory. All roads lead back to JPL. I keep on saying that, and it is super true. Strains of bacterial species Enterobacter bucandescens isolated from the International Space Stations were studied. 13 strains of E. bucandescens, a bacterium notorious for being multi-drug resistant, were isolated from the ISS. Study findings indicated that under stress, the ISS isolated strains were mutated and became genetically and functionally distinct compared to the Earth counterparts. The strains were able to viably persist in the ISS over time with a significant abundance of E. bucandescens coexisted with multiple microorganisms and in some cases could have helped those other organisms survive. Publication impact. Closed human public environments such as the ISS are unique areas that provide an extreme environment subject to microgravity, radiation, and elevated carbon dioxide levels. Any microorganisms introduced to these areas must adapt to thrive. By delving into microbial dynamics in extreme environments, this research opens doors to effective preventative measures for astronaut health. And I'm pretty sure that we had talked about this. If not, I'm sure it will come up, and we will. I'll find it sometime and put it on there. Because it's related. If we haven't talked about it, I'd be surprised. But I will make sure that we talk about it again if we haven't. And that what I'm talking about is killing bacteria with magnets. And so I don't want you guys to be super upset about some multi-drug resistant bacteria on the International Space Station. A, it's on the International Space Station, so that's really far away. And B, they have really strict cleaning stuff there, so that's why they're even detecting this, is because they're testing for this sort of stuff and they're like knowledgeable of looking for this. They're going to turn this into a win for us by being able to study it. But C, there's a bunch of technologies that kill bacteria. There's light, there's chemicals, there's magnets now. And I think I know now that I say that, I know for a fact that we've talked about magnets and like healing stuff. So yeah, we will just move on from that and go into one of the things that I had promised to talk about last week was the professor group that had said that they had discovered a room temperature superconductor that was graphite last year, and now it's coming out in the last couple of weeks that there's some possible scientific fraud involved, which is very interesting. And it's almost like, is he getting smeared? Is because they're saying that it's not true. So was it just a mistake, or are they trying to attack their credibility? Because the plot thickens. I mean, it's already pretty thick right off the bat, but here we have March 5th, which isn't that long ago. This has been evolving, and I didn't want to just jump right into it and report it right away because of what I'm about to explain right now. Because, as I've said before, I try to be very careful when presenting information to you guys, especially on this cutting edge type of stuff, breakthrough technologies, breakthrough organizations, and concepts, that you got to make sure it's not fake, and you got to make sure that it's credible. And it's very hard to do at the beginning, because that's what we try to do on Inspired Earth. And by we, again, it's the audience and me, that we as listeners and learners are trying to get some fresh information, as unbiased as possible and to the truth. And here we have this group that keeps on coming up with these crazy discoveries. A group of Korean researchers known for its controversial claim of creating the room temperature superconductor LK99 has newly unveiled its discovery of a new superconductor. At American Physical Society in Minneapolis, a professor of PCPOSOS, a material that researchers claim to be a room temperature and normal pressure superconductor, according to postings on X, formerly known as Twitter. The presentation included claims that PCPOSOS exhibits zero resistance, the Meissner effect, and partial levitation on a magnet. The Meissner effect refers to a superconducting state with zero electrical resistance and no magnetic fields from its interior. He showed a magnified photo of a sample that levitates and floats completely above the magnet in certain situations, and released a video clip of an experiment on levitation conducted at a lab called CSTL. But the researcher did not disclose detailed information about the experiment on the lab. Kim said he will publish the synthesis method for the PCPOSOS on ARXIV, an online preprint repository for scientists, later in the day. However, Korean scientific community remains cautious, emphasizing the importance of rigorous verification processes before accepting Kim's claims, which is very good. This is extremely good because of their track record. However, it doesn't mean that, just because they've been accused of something doesn't mean that they're guilty. I will say that. They got accused of having fraudulent stuff, but who knows? That could just be a smear campaign. So it doesn't mean that the society has accepted it or that it has gone through the approval process. President of Korean Society of Superconductivity and Cryogenics added that the presentation at an APS session does not equate to global academic approval. He emphasized the necessity for the independent verification to establish the credibility of Kim's findings. The unveiling of the superconductor came after a group of Seoul-based researchers posted papers on an online archive last year, claiming they had developed a superconductor that functions at ambient pressure and temperatures below 400 Kelvin or 127 degrees Celsius. Superconductors are one of the most sought-after materials by science and technology researchers, through which electricity can move without encountering any resistance. The creation of a room temperature semiconductor would significantly reduce, I'm pretty sure they meant superconductor, reduce the energy costs of electronics, with a wide variety of everyday applications, from MRI machines to superfast maglev trains. but their use is highly limited because superconductivity can typically only be achieved at very cold temperatures or high pressures. Since the paper was released, the LK99 superconductor has won global attention and stirred up controversy whether or not it's real. The international and local science community has remained skeptical about the validity of LK99, prompting efforts to verify its room temperature ambient pressure capabilities. So, this is like the second one. The title is Korean researchers unveil a new superconductor. So they haven't even fully assessed the other one, and now we're on to this one. And it seems like they're putting everything out there. The only thing I would be wary about is if they're trying to collect a bunch of money. But I haven't read anything about that. I haven't read anything about them getting a bunch of investors and trying to just run away with the money. That is obviously what we're like worried about as the scientific community and a bunch of learning people. We're worried that we're gonna get taken for a ride here. So they really need to assess the previous one and start assessing this one. And I don't think this is the same one that was actually approved by the global one, the one that was worked on by the Rome and the Japanese and MIT and like a different one. That I think that was a different superconductor. So we've talked about at least 3 just so far right now. So here we have some news. April 2nd, 2024. Is Planet X/Planet 9 real? Although they may sound like science fiction worlds, Planet X and Planet 9 are both hypothetical planets that have been thought to exist in our solar system. Since the early 1900s, astronomers have been searching for a planet that could be responsible for perturbations in the orbits of other objects in the outer solar system. In the early years of the search, the mystery world known as Planet X. In the early years of the search, the mystery world was known as the Planet X. In the new millennium, the hunt for a 9th planet was renewed, this time under the Planet 9. To this day, a hypothetical Planet 9 has remained possible yet elusive. And it's funny how we constantly have to rename stuff after making fun of it. But why might a 9th planet exist? Astronomers think there may be a 9th planet because it could explain strange movements observed in other objects in the far reaches of the solar system. This kind of hypothesis isn't new. Neptune was discovered in 1846 after astronomers searched for a planet that could explain some of Uranus' orbital irregularities. Because planets are very large, they have strong gravitational effects on other objects in the solar system. As such, when looking for an explanation for large perturbations in an object's orbit, a planet-sized body is a likely explanation. In the early 1900s, astronomers began searching for a planet that would explain irregularities in the orbits of Uranus and Neptune. Prominent astronomer Percival Lowell was a leader in the search and predicted that this unknown world would be a gas giant comparable in mass to Neptune. He named it Planet X. Although Lowell died before succeeding in his search, one of the successors, Clyde Tombaugh, used Lowell's observatory to discover Pluto in 1930. And they have a picture here. It says photographic plates taken 6 days apart with a 13-inch telescope, which Clyde Tombaugh discovered Pluto. The arrows point to the position of Pluto on each of the 2 days. And wow. It's just like a dot has moved and they've discovered a planet. It is a 1930s picture, so I mean, I was kind of expecting it to be a dot, but it is kind of funny. As our understanding of Pluto grew through the further observations, it became clear that it wasn't massive enough to affect the orbits of the ice giants. And when Voyager 2 flew by Uranus in 1986 and Neptune in 1989, scientists learned that there wasn't actually any need for Planet X to explain their orbits. We just hadn't understood the planets themselves well enough initially. The story behind Planet 9. Although Pluto was indeed the ninth planet until it was reclassified as a dwarf planet, it isn't what astronomers today call Planet 9. This term was introduced by astronomer Konstantin Batygin and Michael Brown of the California Institute of Technology. Batygin and Brown proposed Planet 9 as an explanation for the peculiar orbit observed among a group of small icy bodies in the Kuiper belt called extreme trans-Neptunian objects, ETNOs. Some ETNOs have orbits that are oddly tilted and tend to cluster together as they get closer to the Sun. In 2016, Batygin and Brown published a paper in the Astronomical Journal proposing some characteristics of a planet whose gravitational pull could explain these strange orbits. They called this hypothesized world Planet 9, because we can't call it Planet X. Batygin and Brown's models suggest that Planet 9 may have a mass about 10 times that of Earth and be similar in size to Uranus or Neptune. The predicted orbit is about 20 times further from our Sun on average than Neptune, taking between 10,000 and 20,000 years to orbit. Neptune, by comparison, completes an orbit roughly 165 years. Has Planet 9 been found? Although the hypothetical ninth planet has not been directly observed, the search continues. Batygin and Brown's team, along with other researchers, have used their computer simulations to refine their search. By simulating objects in the outer solar system, plugging in varying parameters for a potential ninth planet, and then letting simulated gravity run its course over billions of years, the team can see whether that particular Planet 9 affected those objects in a way that matches how ETNO's orbits have been perturbated. These simulations might have ruled out about 78% of the sections of space Planet 9 might have been lurking. That's interesting that they're using the AI to know where to look. Planet 9 hunters are now combing through observational data from advanced telescopes to look for signs of a large planet in the remaining search area, including NASA's Transiting Exoplanet Survey satellite. Brown and Batygin also plan to use data from upcoming Legacy Survey of Space and Time, Skies Survey from Vera C. Rubin Observatory in Chile. So who gets to name the new planet? If a ninth planet is someday discovered, the person who discovers it will get to choose its name, as long as the name is approved by International Astronomical Union. Planets have traditionally been named after mythological Roman gods. The possibilities for naming features on its surface, like mountains, craters, and even individual rocks, could be much more creative. For example, mountains on Saturn's moon, Titan, are named after mountains from Middle-earth, the fictional setting of J.R.R. Tolkien's Lord of the Rings novels, and craters on Mercury are named after influential artists, authors, and musicians. The existence of an undiscovered planet isn't the only possible explanation for the movements of ETNOs. Gravitational effects could be caused by clusters of objects rather than just one big one, or by a very distant small black hole, which we might also just not know enough about ETNOs to really know what's happening with them. This was the case of Uranus and Neptune back in the early days of Planet X speculation. Whatever the case may be, we'll only find out whether Planet X/Planet 9 exists is by studying space. Here we have a very, very interesting one. This is novel DNA mechanism in memory discovered April 19th, 2024. Researchers identified a novel mechanism that impacts memory formation through changes in DNA structure, specifically quadruplex DNA. Their study reveals that G4 DNA accumulates in neurons, dynamically influencing gene activation and repression crucial for long-term memory. By employing CRISPR technology, the team demonstrated that DNA helicase DHX36 directly regulates these G4 DNA structures in the brain. The discovery not only changes our understanding of the DNA's role in memory, but also new avenues for investigating memory-related disorders. Key facts. The study provides the first evidence of G4 DNA presence in neurons, highlighting its functional role in regulating memory-related gene expression. Researchers used advanced CRISPR-based gene editing to pinpoint how G4 DNA structures are regulated in the brain, finding a crucial role for DNA helicase DHX36. The studies suggested that DNA structure, beyond just its sequence, plays a critical role in how experiences are encoded in the brain, potentially impacting treatments for memory-related conditions. Okay, that is, I know this is a lot. You might have to go back and re-listen to this, because I have to go back and reread this stuff. It's not, I'm pretty sure most people have to go and reread this stuff. But do you remember that DNA is just proteins? And we talked about how proteins are constantly moving, a lot of them. And here it says how this protein that is constantly moving impacts its role in how experiences are encoded in the brain. Is that not fascinating and bizarre and strange that we have? And proteins are just made up of bonds of chemicals. So we have these weird vibrating chemicals that make up our experiences, and that's extremely reductionist. That's only part of the equation. That is not the whole equation of consciousness, but we're talking about encoding in the brain of memory, and the fact that the DNA structure affects how memory is encoded is very bizarre, potentially impacting treatments for memory-related conditions. An internal collaborative research team, including scientists from UQ's Queensland Brain Institute, QBI, has discovered a novel mechanism underlying memory involving rapid changes in a specific DNA structure. The team found that G quadruplex DNA accumulates in neurons and dynamically controls the activation and repression of genes underlying long-term memory formation. In addition, using advanced CRISPR-based gene editing technology, the team revealed the causal mechanisms, or causal, yeah, I was right the first time, causal mechanisms underlying the regulation of G4 DNA in the brain, which involves site-directed deposition of the DNA helicase DHX36. The new study, published in the Journal of Neuroscience, provides the first evidence that G4 DNA is present in neurons and functionally involved in the expression of different memory states. The study, led by Dr. Paul Marshall at Australian National University and QBI, and a team of collaborators from Longko University, Weizmann Institute of Science, and University of California, Irvine, highlights the role that dynamic DNA structures play in memory consolidation. DNA flexibility. For decades, many scientists considered the topic DNA to be solved. DNA is widely recognized as a right-hand double helix, which changes to this structure only occurring during DNA replication and transcription. This structure contains two strands of nucleic acid featuring the four bases: adenine, thymine, guanine, and cytosine, which pair together to form rungs of a DNA ladder. We now know this is not the complete story. QBI's Professor Tim Brady explains that DNA can assume a variety of conformational states that are functionally important to cellular processes. DNA topology is much more dynamic than static right-hand double helix, as presumed by most researchers in the field. So he's saying most researchers don't know what they're talking about with the shape of DNA and the role. There are actually more than 20 different DNA structure states identified to date, which is probably barely any of what they actually are capable of, I would just guess, each potentially serving a different role in the regulation of gene expression. In the new study, the team has now shown that a significant proportion of these structures are causally involved in the regulation of activity-dependent gene expression and required the formation of memory. Although epigenetic modifications have a well-established association with neuronal plasticity and memory, to date, little is known about local changes in DNA structure affect gene expression. G4 DNA accumulates in cells with guanines fold into stable 4-stranded DNA structure. While there is evidence for the role that this structure plays in regulating transcription, prior to this study, its involvement in experience-dependent gene expression had not been explored. G4 DNA transiently accumulates in active neurons during learning. The formation of this quadruplex structure takes place over milliseconds or minutes, at the same rate of neuronal transcription in response to an experience. The G4 DNA structure can therefore be involved in both the enhanced and impairment of transcription in active neurons, based on their activity to enable different memory states. This mechanism highlights how DNA dynamically responds to experience and suggests that it has the capacity to store information not just in its code or epigenetically, but structurally too. So G4 DNA regulates memory, and it can do that through its structure, not just by changing the way that it's encoded. Like literally its shape is, it sounds like it was even being changed from our experiences. The formation of this quadruplex structure takes place over milliseconds or minutes at the same rate of neuronal transcription in response to an experience. So yeah, that's like saying that your DNA is changing based on your experiences in the moment. That's like, it's crazy, but it kind of makes sense in a way, but that's very counterintuitive to what we have like thought. It takes a long time to process that. I'm processing it right now. It's almost jarring to hear that your DNA can be changed in milliseconds or minutes at the rate of neuronal transcription in response to an experience. It's bizarre, and really, for me, it makes me reflect of how all of humans need to take care of themselves better with that in mind. Like if you are having a bad day, apparently that can influence your DNA. So it might be worth it to try to have a good day. That's pretty interesting. And here we have it going into fear, which it just gets deeper and deeper, man. Who would have thought that DNA would go into psychology so quickly? But it does. This is a really good study for it to, it's a very deep study for it to go into all of this. The extinction of conditioned fear is a behavioral adaptation that is critical for survival. Fear extinction relies on forming new, long-term memories with similar environmental elements to compete with and take over the fear-related memory. The formation of long-lasting extinction memories depends on coordinated changes in gene expression. Professor Breedy said it is now evident that activity-induced gene expression underlying extinction is a tightly coordinated process. Wow. This process is dependent on temporal interactions between the transcriptional machinery and a variety of DNA structures, including G4 DNA, rather than being determined solely by a DNA sequence or DNA modification, as so often has been presumed. The discovery extends our understanding of how DNA functions as a highly dynamic transcriptional control device in learning and memory. That's crazy. That's just nuts. They actually just came out with something in the last like 24, 48 hours that hit the news, talking about something that was very similar to this. I don't have time to just read the whole thing on it, so I'll just quickly say that they had been studying the spine of some mice, and they figured out that the spine can be trained without the brain, and that the DNA will hold the memory of certain actions, and that you can teach the spine without the brain, which is kind of freaky. But if you think about it in like a paraplegic type of thing, that people have spine injuries, and if you could teach your spine how to walk, like that would obviously be like a machine basically doing that for you. Like you can't do that, but like a machine can electrocute those neurons that need to be electrocuted and do the things that need to do and activate the DNA. Well, it was talking about DNA delivering some of this stuff in the spine, and that is how they discovered that the spine had memory. Because how can they store how can a muscle or a neuron gets zapped by electricity and then remember that for days? Because that's how the study was working, was they had been doing this for several days. So yeah, I don't want to get off on it too hard. I'll read it sometime. I'll skim over it, but I just wanted to mention that this even goes beyond the brain, and that they're already looking at healing people from spine injuries with this, like immediately. That's what they were talking about. And here we have people talking about fear. So imagine us being able to treat PTSD or anxiety or any number of things. You could probably even throw like substance abuse into that. Most likely could be treated through DNA treatment, which is actually crazy to even say. It's just bizarre. 1 week after another, the technology just keeps on going and blowing my mind, blowing all of our minds. And thank you guys for listening. I appreciate it. I can't wait to learn some more. I'm sure that you guys are right there in it, and we're all gonna go through it together. We're all in the boat together, learning and traveling through this weird world, and we have love to guide us. And that's basically where we're at right now, is we have love and information and facts, and we're gonna go with that. And I hope everybody's safe out there, and we'll see you again.